rational drug design. All right. So sometimes we'll have examples where this is. Sometimes we'll have examples where this is kind of there's some crossover, there's some overlap. You'll see some similarities, right, between this and high throughput screening. Sometimes high throughput screening provides the foundation for rational drug design. But basically, to put it into a simple explanation, rational drug design is targeted. You're specifically looking for some sort of effect. You're adding it or you're subtracting it, right, from a known compound, or you're trying to fit into a known target. Okay. High throughput screening, to put it in Candy's words, is you're throwing everything against the wall and you see what sticks. All right. So what I have here is two young men fishing, and this is a pool of thousands of compounds, like let's say 30,000 compounds, right? And this is a hit. Okay. Whereas target design, we would start with something that, let's say, would be our lead, and we'd be modifying it. Or we would start with a target, right, and we would be trying to find something that fits specifically into that target. Okay? All right. So, we need some sort of information to get started, right? So this is what I was talking about, information about the target, or information about other molecules that have a desired effect. Okay? So for example, we can show morphine, right, and add different functional groups, right, that give us a desired additive effect, or subtract something, maybe reduce its half-life in the blood or increase it, depending on what we want to do, right? Okay. So there are two types of drug design. Okay? We have structure-based and ligand-based. Okay? With structure-based, we have a structure of a target. We need that for structure-based drug design. Okay? And we're trying to design a drug that is going to fit into the structure of the target. Right? And like Candy said, the target could be anything. Right? Very frequently it's a protein, right? But it could be any kind of biological entity. Okay? Right. Now ligand-based, we have already something which is a kind of a basic molecule. Right? You can say a lead, right? Or some idea of a class of molecules that have a desired effect. Right? And we're trying to improve upon them. We're trying to add functionality to them, or we're trying to make sure that they're going to work well in the human body. Okay? So these are the two types. Sorry, I don't want to get you with my laser. So structure-based and ligand-based. Okay? Now, how do we obtain information about the structure of targets or ligands? Right? So, can anyone just throw out a technique? X-ray. X-ray crystallography. Someone else. There's another main one. Okay? Anyone? NMR. Perfect. Well, you're a lecturer, so I'm not surprised that you know.
You better get that one right.
You better get that one right. Okay. So that's exactly right. So X-ray crystallography is the one I put as number one. Okay? NMR is another very important one. And if you don't have, or you can't get, crystallography data or NMR data on your target, you can also do something that's called homology modeling, which I'll explain later, using a computer and some other pieces of information. Okay? So, other helpful techniques, electron microscopy. Sometimes you'll see really amazing papers where they'll show you a protein complex. They'll show you something like a complement protein or an antibody binding to its target, and they'll show you what it looks like with an electron microscope. It's really cool, but it's hard to do. Okay? Pull-down assays. If you want to look at how different things interact, like for example a protein-protein interaction or a protein-DNA interaction, we're talking about pull-down assays. Okay? But that's not the focus here. Another thing, analytical chemistry, basically liquid chromatography and mass spectrometry. So with mass spec and liquid chromatography, you can do so many things. And really that's very important for drug design. But we're going to focus on these techniques here. If you have any questions about anything else, just send me an email. Okay? All right. So, this is a case study of rational drug design. Who here has heard of Gleevec or imatinib? One person. Okay. Well, this is one of the most important drugs you will ever hear about. Okay. This is the first targeted kinase inhibitor for cancer therapy. First approved. Okay. It was FDA approved in 2001 as a frontline treatment for CML. And I'll get to that. It's a leukemia. Right? So there was a lead compound that was identified by high throughput screening. Okay? And then, after that, the medicinal chemists took over. Okay? And they began working on this. Right? The reason why here in the structure you have R is that this R group, they found many different compounds where that had this structure and then something here, whatever this R was. Right? Basically, all they did was added a methyl group to change these inhibitors that have cross-reactivity between the target, which is BCR-ABL, right, and these other kinase, protein kinases, which PKC, so protein kinase C, right. Just adding this one methyl group right here, they were able to make selective BCR-ABL inhibitors, right. So selective for this cancer target, okay. Now, then this was basically distribution, metabolism, pharmacokinetics optimization. That's what DMPK stands for. All right. They were able to make Gleevec. Okay. So this is just a short list of the kinases that it binds to. The reason is these kinases are related, right? So there is some cross-reactivity, but one of the reasons why it was very effective for CML, because this is basically the most important molecule. This is kind of the etiology of the disease is based on this fusion protein. Okay? And we'll get to that. We'll get to that later. Okay. So what type of drug design is this? Oh, yep.
Ligand-based?
Yep, ligand-based. Exactly. Exactly right. So we're starting with the ligand, and then we're optimizing it. We're working with it. We're building upon it. Right. We can do this without any knowledge of the structure of the target. And that's how you know the difference. Okay? One example of structure-based design, so the other type, right, is, for example, there was a HIV protease, right, that we wanted to create a protease inhibitor. We know the structure, but we didn't know any natural molecules, naturally occurring molecules that could inhibit that protease. Right? So we built a molecule that could fit, right, into the active site of that protease, and thereby inhibiting it. So that's another example. Okay. But now, CML. So this is chronic, you can say myeloid leukemia. This fusion protein is a result of this, what's called the Philadelphia chromosome. Okay? So we have the ABL gene from chromosome 9 and the BCR gene from chromosome 22. Okay? We have this fusion product, and this is the Philadelphia chromosome right here. Okay? This is a normal blood smear. All right, you see the leukocytes are relatively few compared to the erythrocytes. Okay, and this is a leukemic blood smear. And you see there's a big difference. So through this very simple blood smear microscopy, you can diagnose leukemia. All right. And you can tell that it's from myeloid cells by looking at the morphology of the cells. They're not lymphoid, they're myeloid. Okay? All right. So, these two main methods of learning the structure, right, of a target. Okay? So now we're kind of moving to structure-based drug design. Okay? So we've got nuclear magnetic resonance, all right? And here we have, this is NMR data, all right, of imatinib in orange binding to ABL. So this is where it binds to on BCR-ABL fusion protein. Okay? Now, these magenta parts of the protein are the ones that move upon binding. Okay, so there's kind of a conformational change upon binding, right? And here you can see the pocket, the binding pocket, right, where imatinib fits in. And you can see it's actually a really good fit, right? Okay, so a couple things about NMR. You're working with your target in a liquid, or it could be dissolved crystals, so liquid crystal phase, right? this means you can use what's essentially a natural milieu of the target, and you can capture movement. It's active, right? That also makes it harder to have really good resolution, right? If you've ever tried to take a picture of someone that was moving, it's difficult. It might become blurry. That's kind of what happens here, right? So to compensate for that, we can have really powerful magnets, okay? This is basically what gives us the power to have good resolution with NMR, right? And if you're familiar with how MRI works, this is similar to that, okay? Now, another way you can make this technique be more powerful, achieve better resolution, is to substitute normal atoms with these exotic or rare forms, right? And then, basically what this requires is you express the protein, right, in media that contain these rare nuclei. Okay? Alright? Now, so this technique is really not ideal for large proteins, right? With a really powerful magnet and with these, this sort of incorporation technique that I talked about, you can make it work for larger proteins. But really 20 to 30 kilodalton is where it starts getting a little bit fuzzy, and greater than that is really difficult, okay, with NMR. Now, X-ray crystallography doesn't have this size limitation. Okay? You can have really huge complexes in a crystal and be able to get crystallography data very easily. Right? The hard part is getting good crystals, especially getting good crystals of complexes. All right? For that you need very high purity protein, right? And this binding has to be very good. Okay? Luckily for us, the binding of imatinib to ABL, right, is about 14 nanomolar, right? And that's pretty decent. If we're talking about an antibody, I'd say that's pretty decent for an antibody. So that's really strong binding. Okay? All right. So key difference is that this is solid phase only. It has to be a crystal. So you're not going to see any movement. So the resolution generally is going to be better, right? But you won't be able to see any conformational changes. Okay? All right. This is the gold standard for protein structures. And what I mean by that is if you look at this protein database, or PDB, right, you'll see that of these 70 or so thousand protein structures that are available there, you know, the vast majority of them are from X-ray crystallography. Okay? Okay? And a good number are from NMR, and then there are other techniques. Okay? But these two are definitely the main ones.
If I may add.
Yes.
The protein database is a website. So anyone could go up and take a look at it.
Yep. And you can basically find a protein, look at its structure, open it up in JMOL, which is a program for viewing the structure, make it rotate, make it dance, zoom in and out, highlight the residues you want. Okay? That's a lot of fun. Okay, so from this crystallography data, this was done actually after Gleevec was developed, right? You can look at the crystallography data and say, okay, this moiety on our small molecule interacts with this amino acid in the target, right? And so on and so on and so on. And you can see which residues are important. Where are the interactions happening? Okay? All right. Now, if you don't have any information on the target structure, okay, there's still something you can do, right, to get started with structure-based drug design. All right. Now, you can do homology modeling. Basically, what you have is you have a template protein. Okay, this is a known structure of a protein that's similar, so it's homologous to the protein of interest, to your target. Okay, and you need the sequence of these two. Okay, the known structure and the unknown structure. You do a sequence alignment, right? And this in the computer, in silico, you can make the structural model. Okay. So basically what the computer does is it takes these sequence differences and it grafts it onto our template, okay? Which very often is going to be X-ray crystallography. Okay? All right. So what next? We have computer-aided drug design. All right? This is when we have our structure. We know something about it, or we can use the computer to say, ask a question such as, where is its active site? Where is the, its activity occurring, right? Does it bind ATP? Does it need ATP, right? Maybe we need maybe we can have it bind in the ATP binding pocket and thus block the activity of this protein, as an example, right? Maybe we can find an allosteric site, right? Where we bind and we inhibit, right? Okay, so having a good fit, this is basically the kind of the size and shape of the molecule is a good fit for wherever we want it to bind in our target, right? And then there are the interactions, like when I showed different moieties of the ligand, right, interacting with amino acids of the target, okay? So a couple examples, right? You can have hydrophobic amino acids. Do you remember when we talked about amino acids? Yeah? You can have hydrophobic amino acids binding to kind of a hydrophobic pocket, right, in the target. Yeah? You can see from structural data where are there water molecules, right? And we can displace those water molecules using parts of the small molecule that will have electrostatic interactions with amino acids in that region. Okay? Alright. So after you come up with a design like this, you need to validate. Okay? Because anything you show in silico, it may be great, maybe the next big discovery, or it may not mean anything. Right? You need to validate it using an assay. Okay. So what about biologics? So up until now we've mostly talked about small molecules. Right? You can also use many of the things we talked about for testing biologics. So by biologics I mean something like an antibody, some sort of protein, something that's produced by in or by a cell. Right? Okay.
So would that include like natural products?
So some natural products, they're actually small molecules, right? And that, a lot of antibiotics, they're actually made by an organism, but they're small molecules, right? Okay. So really it's a kind of a size difference, right? Where proteins, right? When we're talking about macromolecules, right? Like an siRNA, that could be like a biologic, right? When we're talking about macromolecules, that's when we're talking about biologics. Right? It really is kind of a size difference. Yeah? Okay? So one of these components of the rule of 5, right, is you want your small molecule to be less than 500 Dalton. Right? Okay? And when we're talking about like an antibody, right, that's 150 kilodalton. Right? So that's much, much larger. Right? Most small molecules there will be less than a kilodalton. Okay? Alright. So now going on to biologics. Phage display. Who's familiar with phage display? Hand in the air. No one? Okay. So basically you use a bacteriophage, which is a virus that infects bacteria, to couple a protein with the genetic material that's used to encode it. Alright? Okay? Then you have this protein that's expressed on the surface, it's displayed on the surface, right? You use it to bind to your target, okay? And then you can select for good binders, right? Elute them out, and then amplify them. How would you amplify them? It's a bacteriophage.
Bacteria too?
You add it to bacteria, they infect the bacteria, they replicate, they lyse the cell, and they're released. And then you have a bunch more where they came from, right? Okay, there you go. And you can repeat this over and over and over again to get very good binders. Okay, so this is kind of screening for binding using a biological system. Right? This is phage display. Okay. Now, for testing vaccines, you can screen vaccines using an ELISpot. Right? This is essentially an ELISA that's using cells. Okay. When we stimulate a cell, like for example a T cell. Right? Here we have an antigen-stimulated T cell. Right? It's going to be secreting a cytokine. Right? We have a capture antibody at the bottom of our plate. All right? And it's capturing the cytokine. We've washed away the cells, but the cytokine remains. Okay? Then we have a detection antibody, right, which is going to give us our signal. Okay? And here it's a chromogen, so it would be for a colorimetric assay. Right? But it can honestly be anything. Right? Something linked to this detection antibody that's going to give us our signal. Okay? For antibodies, when we talked about antibodies, we talked about the CDRs. And so there are 3 CDRs, right? On each chain, the heavy chain and the light chain, right? And they come together to kind of grasp the antigen that the antibody is binding to, okay? And so these are the 3, so 6 total CDRs, right? You can use homology modeling to figure out, well, what is this antigen binding portion of the antibody? What is its structure? Okay? So you have the sequence. You don't know the crystallography data of the antibody that you're interested in, but you have its sequence. Right? And you can use this computer-aided technology, right, to find out more or less what its sequence is going to be like. So here's an example where in yellow, this is the actual sequence. This is the actual structure of an antibody. And in this sort of pink color, right, this is what was the result from homology modeling. Right? It's quite good. Right? It's quite close. Okay, so some final thoughts. Generally in drug design, you need to start somewhere. So ligand-based, you need to know something about the ligand, its structure, its activity, something like that, right? For structure-based, we're talking about the target structure. And without that, you can't get started with structure-based design. Okay? And then homology modeling, you need the sequence of the target, the sequence of a homologous protein, okay, and the structure of the homologous protein. Okay? And then you can, in your computer, predict the structure of your target. Okay? Okay. So, Gleevec, huge benefit to patients. Okay? So really it was a blockbuster drug. Right? It's a drug design success story. Right? The previously available treatment, interferon, right, was basically very, you could say, inadequate. Right? Gleevec kind of blew everything out of the water, completely shifted the paradigm for CML therapy. Okay? All right. It started a new class of cancer drugs, kinase inhibitors. Now, if you hear about cancer, you learn about cancer therapy, you've probably heard about cancer therapies that involve kinase inhibitors. Right? And a lot of the blockbuster drugs in the past, also in the future there will be, are antibodies. But now kinase inhibitors are rivaling monoclonal antibodies as these top targeted therapeutics. Okay? And here's a table. Right? Number 1 right now is Humira, right? Which is an anti-inflammatory antibody. Right? In terms of targeting cancer, the top one is Rituxan, or rituximab. It targets CD20 on the surface of B-cell malignancies. Okay? Right? These are the top earners. This is in millions of dollars over here. Okay? So Gleevec is down here, right? But remember, it was FDA approved in 2001. Okay? So it's still on this list because it's been approved for the treatment of other diseases, right? Like these gastrointestinal stromal tumors. Right? But really, if you think about it, this is a huge financial boon to the company that developed it, Novartis. Right? And it's been a huge benefit to the patients. Okay. So, with that, I want to say to all of you, thank you for listening, and good luck discovering the next Gleevec. Okay.
Question?
Yes?
So I feel like a lot of these assays look at like affinity, like a
Yes.
receptor affinity to the ligand.
Yes.
But like, what about potency? Like, what if it has low affinity, but this drug is very, whatever, potent?
Usually if it binds very well, that means that it's selective, which means that it will have, let's say the off-target effects will be less, right? If it has good binding affinity. Usually that's a first step. You should not move on with the drug unless you test it on cells, right? And so with Gleevec, for example, they found out that an inhibitory concentration for CML cells, okay, was 0.4 micromolar, right? Which is pretty good for an IC50. Right? So that gives us an indication also of its efficacy against cancer cells. Right? So they're both very important pieces of information. Right? Generally, you want to know how well does it bind to the target kinase, how well does it bind to the other kinases. Right? If it doesn't bind to the target with an IC with a binding affinity that's somewhere in the physiological range that we could expect, right, of a patient taking the drug, then it's not going to be useful.
Yeah. So you look at affinity first, and then
Usually that's easier because you just put the whatever the target is and the drug in the plate, and that's it. With cell-based assays, it's still very doable and can very well be high throughput, but it might be a little bit more complicated.